Hello and welcome to another video of Vidhai. I'm Ryan, a volunteer from Exams Vidhai, where everything is possible. In this video, we'll be learning about breeding and biotechnology for the exam NTSE. So let's take a look into the introduction to breeding and biotechnology. India's population is likely to reach about by 2050 it's 1.7 billion. At this rate, only 60% of the country's food demand will be delivered at the time. So how can we make sure that everyone gets appropriate nutrition through agriculture? And this is made possible through plant breeding and animal husbandry. Plant breeding is the scientific method of reproducing plants, more or less agriculture of plants, where it is mainly focused on specific traits of the plants which we desire. And same goes for the animal husbandry, where we make use of the genomes and other phenotypes to make sure we get a better quality of milk and meat. So let's take a look into the plant breeding. In this video, we'll be focusing on plant breeding. In the next video, we'll be focusing on animal breeding. So in the plant breeding, we have the green revolution, where we have 4 different subtopics of high yield, disease resistance, insect and pest resistance, improved nutritional qualities. These are different types of aim that the green revolution has to provide. Before looking to the green revolution, we'll be looking into crop improvement, and then we have the methods of plant breeding. We have 4 main methods of selection: polyploid breeding, mutation breeding, and hybridization. In selections, we'll be focusing on the artificial ones, that is the mass, pure line, and clonal selection. So crop improvement. Modern agriculture practices are activities carried out to improve cultivation of plants. So we have sowing, we have fertilizers, we have harvesting, we have irrigation. Modern agriculture makes sure these fundamentals are intact, and including it, we be looking into further scientific methods and basic methods to improve the crop yield that we get from our harvesting period. So the aim of crop improvement is to develop improved crop varieties possessing higher yield, better quality, and resistance to disease and shorter duration. So let's take a look into the green revolution. The green revolution is the process of increasing food production through high yielding crop varieties and modern agriculture techniques. This was made through Dr. Norman E. Borlaug, American agronomist, and is also called as the father of green revolution. He has also received a Nobel Prize in 1970. So what does the green revolution focus on? The green revolution focuses on high yield, disease resistance, insect and pest resistance, and nutritional quality. So let's look into the better yield. So the major challenge that India faces during post-independence period was having enough food production for the growing population. So we need better yield and better quality of the crops that we grow. Here are some few examples of some better yielding crops. Sonalika and Kalan Sona are semi-dwarf varieties of wheat which are high yielding and fertilizer responsive, from Mexico, that is. IR8, which is called as miracle rice, is a high yielding semi-dwarf rice by the International Rice Research Institute in Philippines. In 1966, this was first introduced in Philippines and India, that is IR8. It was a hybrid between the Pita from Indonesia and DGWG from China. So what is hybrid? We'll be looking into the further slides. So the next subtopic of green revolution is the disease resistance. So plant disease are caused by pathogens like virus, bacteria, and fungi. So we need to develop a crop that is resistant to these diseases. So this tabular column is also in your textbook, so don't worry, you can also refer that. But for the quick review, for wheat, there's a variety of wheat called as Himgiri, which is resistant to leaf and stipe, rust and hiltburnt. We got cowpea, which is a variety of Pusa Komal, which is resistant to bacterial blight. So next we have the similar thing but for insects and pests. So you have a crop of flat beans, and the variety could be of flat beans of Pusasem 2 or Pusasem 3, which is resistant to insects like leaf hoppers and fruit borers. We got lady's finger variety where it is resistant to shoot and fruit borers. Next we have the nutritional quality green revolution. So what does nutrition quality mean? It is increasing the protein quality and quantity, oil content and mineral content of the growing crop. Why do we need to focus on nutritional quality of a crop? This is because to tackle the problem of undernutrition and protein malnutrition among the human population, which is actually a major health problem which has been receiving much focus throughout the world. To combat these conditions, humans and animal health are to be determined by nutrition quality of the fed crop. We got biofortification, a method of improving nutritional quality. So what does biofortification means is the scientific process of developing crop plants enriched with high levels of desirable nutrients like vitamins, proteins and minerals. Some of which examples are Atlas 66, a protein rich wheat, and then you got Proteina Shakti and Ratana lysine rich maize. We got iron rich fortified rice varieties as well. Now for the methods of plant breeding, this is the next subtopic in the plant breeding. We got mainly 5 important plant breeding methods. So we got introduction to new varieties of plants, selection, polyploid breeding, mutation and hybridization. The first one is rather minute, so we'll finish it off in this slide itself. It's the process of introducing high yield varieties of plants from one place to another. Such plants are called exotic species. So what do we do is bring a high yielding plant from one place to another place and introduce there in that agriculture. So all the other plants will support the high yielding exotic plant, and the exotic plant in turn will return its characteristics of high yielding nature. So the next is selection. Selection is the oldest method in plant breeding. Individual plants or group of plants are sorted out from a mixed population based on morphological characters. So there are 3 types of selection: mass selection, pure line and conal selection. So mass selection is really easy to understand. Like from this picture you can understand what's happening. So you select one particular shape from this group of shapes, and then you place it in the empty fields like this, and then when you keep on doing this iteration for 8 to 9 generations or 7 to 8 generations, you will in turn get a field only with that shaped crop. So seeds of best plants showing desired characters are collected from mixed population. This first step is called mixed population, and this thing is carried out for about 8 to 9 generations, and thus we'll get the desired crop variety from mass selection. Pure line selection is more or less confusing, but it's a self-breeding process. If you had heard about reproduction in plants, you might have heard about the terminology of self-pollination. So there, if it's self-pollination in flowers, here it is self-breeding. So self-breeding is not only focused on flowers but also in plants as well. So a single individual will give more than one offsprings, and those offsprings will give more than one offsprings, which means its power to power is exponentially increasing. Thus, you can bring out one whole field of crops from just one plant itself, and this is, since it's from only one plant, it's called pure line selection. So as you can see, a large number of plants are selected from a self-pollinated crop and harvested individually. So that's the important thing we need to know. Clonal selection is more or less same as pure line selection, but clonal selection is the next step of pure line selection. Pure line selection you are allowing to grow, but clonal selection is that you're taking out specific clones from the pure line bred selection. So a group of plants produced from a single plant through vegetative or sexual reproductions are called clones. So clones are the product of pure line selection. All the plants of the clones are similar in both genotype and phenotype. You will come across this word genotype and phenotype more likely than ever. Genotype is something which is in genes, and phenotype is the physical appearance. Selections of desirable clones from mixed population of vegetative propagative crop is called as clonal selection. So we are taking a pure line selection and then mass selecting it, and these two processes are together is called as clonal selection. Next we'll go into polyploid breeding, which is kind of confusing, but the more you read into it, the more understandable it gets. So sexually reproducing organisms have two complete sets of chromosomes in their somatic cell. This is called diploid, and diploid means you have somatic cells and then you have gene cells. So somatic cells has somatic chromosomes and gene chromosomes. So an organism having more than two sets of chromosomes is called polyploid. So us human beings are diploid because we have two sets of chromosomes only, and in such condition called as chromosomes, it can be induced by physical agents such as heat or cold or x-rays and chemical agents. The achievements of polyploid breeding is that watermelon seeds and banana seeds are triploid, meaning that they have more characteristics than before. This could also in turn mean as more nutritional qualities or more disease resistance qualities, as well as insects and pest resistance. A TV29, which is a triploid variety of seed, that is 3N only, with larger shoots and drought tolerance. And then you got triticlate, which is a hybrid of wheat and rye, which is a hexaploid. Now we'll go look into the next part of the plant breeding, mutation breeding. Mutation breeding is, before we go into mutation breeding, we need to know what mutation is. So mutation is defined as the sudden change in nucleotide sequence of a DNA in an organism. So this is caused by physical mutagens and chemical mutagens. So the physical mutagens are like X-rays, alpha, beta, gamma, UV and temperature, which induced mutations are called as physical mutation. Now radiations that you might have more or less associated with atomic bombs, which are more or less right, and atomic bombs produce gamma rays and UV rays and X-rays most of the times. So when you are exposed to radiations, right, for prolonged period of time, it is bound to change your genetic code in your somatic cells, meaning that your entire phenotype could change based on your genotype. Now we have chemical mutagens like mustard gas and nitrous oxide, right? So why we prefer chemical mutagens over physical mutagens? Because physical mutagens not only harms us, but even though we are targeting plants, it will harm us as well. But chemical mutagens are more or less a one target method, so we use chemical mutagens. Using chemical mutagens, we have groundnuts with thick cells, and then we got Automia 2 rice with saline tolerance and pest resistance, and then we got wheat from Sonara 64 using gamma rays. So the next step in plant breeding is hybridization. Hybridization, you might have come across this again in your reproduction class for plants, where when you cross pollinate or you cross select between two different traits of a plant, they give a hybrid kind in the end. So the process of crossing two or more types of a plant for bringing the desired character into one progeny is called hybrid. So the first hybridization experiment was Tetriclade. It is the first man-made cereal hybrid, and it's a hexaploid Tetriclade. The selection of crop The cycle of crop raising and selection continues till when the desired characters are finally obtained. This process is through hybrid vigorously, and it's a long process as well, but the results are in the end futile. But the results in the end are profitable. After we get the hybrid plant, we can do pure line selections or clonal selections. So for this video, I would like to end with the practice questions. So practice question 1, which method of crop improvement can be practiced by a farmer if he is inexperienced? Himgiri developed by the hybridization and selection for disease resistance against rust pathogens is a variety of what? The miracle rice, which saved millions of lives and is celebrated its 50th birthday, is what? So I'll give you some guys time to solve these questions on your own. You can pause this video and go ahead. Now I'll be discussing the answers to this. And the answer for the first one is clonal selection, because it's the most easiest one, and since clonal selection is the mix of both mass selection and pure line selection, this is more or less the most efficient and easier method to use. Now the himgiri is a variety of wheat. That's basically you'd need to know in order to answer this question. All the other crops don't have the variety of himgiri, but wheat has a variety of himgiri which is resistant to rust pathogens against disease. The miracle rice that we have learnt in this previous slides is IR8, and other are not the rice. This is called as the miracle rice. You can have this ideology of, since you have only learned IR8 and not the rest, IR8 should be the answer, because other options not being taught in the textbook is probably not the answer. This is also one of the strategy that you can use when answering in NTSE. And that concludes the first video of the chapter Breeding and Biotechnology. All the extracts are from the textbook and only pictures are from the textbook and Google Images. Remember to go through the glossary and points to remember behind the chapter of the textbook. Please do like, share and subscribe, and there is a second video to this, and check it out. In the second video, we'll be learning about the plant breeding and biotechnology in detail. Thank you once again.